Cheers. All right, so I guess I don't really need to explain the title, but maybe I will anyway. We all know that being cryopreserved is the second worst thing that can happen to you. Actually, I think now it's officially the third worst. You know, the second worst is mind files, which are like a bit of an approximation to being preserved. But yes, so of course we don't want this to happen. But one of the things that actually led me to be to find it so easy to understand the rationale for cryonics when I first started hearing about it, maybe 12 or 13 years ago, was that the process of reviving somebody, and of course of repairing all of the damage that has been done both during the process that caused them to become legally dead in the first place and during any cryopreservation process, that process of repair and revival seems to me to be very likely to have a great deal of similarity to the kind of approach that I have been developing and advocating since about the year 2000 for people, as I say, whose hearts are still beating. So it was kind of a natural extension of the same thing. And so what I'm going to do today is work in the opposite direction, addressing obviously people who all know a good deal about the nature of cryopreservation and potential revival, especially after the excellent talks we've heard, and to talk about the kind of work that we might be able to do to avoid having to be cryopreserved in the first place, and maybe the similarities will be apparent. What SENS Research Foundation does, of course, is we work on trying to repair the damage that the body does to itself during life, and we try to keep people youthful as a result. But I want to step us back a little bit from there first, in order to put that in context, to explain why this is an approach to combating aging that seems promising, and how it compares and contrasts with other approaches to keeping people healthy. And I think I'd like to start here with something that we often forget, actually, but it really is quite paradoxical if you think about it, that we have been so spectacularly less successful in combating the diseases and disabilities of old age than we have with regard to infectious diseases. Very primitive elementary methods, such as realizing that hygiene was a good idea and vaccines and antibiotics and so on, have been capable of virtually eliminating most infectious diseases in the industrialized world. And yet our progress against the diseases of old age has been really very modest indeed. So we have to ask ourselves why. And I think that's a good way to lead into a definition of aging, which is a very useful thing to have. A lot of the most unhelpful comments by dignitaries in the field of the biology of aging are made as a result of a failure to incorporate a proper, rigorous definition of what they're talking about. And so I'm going to start by doing that. I believe that a definition of aging that's mechanistic, that says how aging happens, is the right place to start. So what I point out here is that aging is a process that occurs as a result of the body's normal operation. And another really important aspect of this definition is that it demystifies aging. This is something that I probably don't have to belabor with this audience, but certainly when I speak to the general public, it's very important to get across to them that aging is not a mystery, the way that they have been brought up to think it is. Aging of the human body or of any living organism is really, in essence, no different than aging of an inanimate object like a car or an airplane or whatever. And the reason it's essentially no different is because it's a fact of physics, never mind biology, that any machine with moving parts is going to inflict damage on itself throughout its existence as a side effect of the way it works. It's also a fact that any machine, living or inanimate, is going to be built to be set up to tolerate a certain amount of that damage. And sure enough, the human body is set up to tolerate quite a lot of that damage, which is why the typical performance, both mental and physical, of, let's say, a 45-year-old person is not very different from the typical performance of a 25-year-old person, even though, of course, they have a very considerably smaller remaining longevity. So this is a very straightforward definition. It's very down to earth and it's very useful. But it's not the kind of definition that is actually adopted and generally acted upon in society at large. And this is something we need to change really quite soon, because the misconceptions that are so prevalent about aging in society cost us a lot of money and a lot of time. One way of describing those misconceptions is here. We have a good idea in our minds of the various ways in which people can be sick, and we can classify them into these 4 categories here. We can talk about infections, communicable diseases. We can talk about rare inherited diseases that are caused by problems in your genes. We can talk about the diseases and disabilities of old age, which we're all very familiar with, and of course, which dominate the medical budgets of the industrialized world these days. And then people have this popular idea that there's this completely separate thing called aging, which is this kind of nebulous non-disease phenomenon that consists of these various non-specific things like sarcopenia, that's the loss of muscle mass, or decline in function of the immune system, things that we don't call diseases. And there's this big, massive black line between the two, such that most people don't really even think in terms of the feasibility, even in principle, of developing medicine to actually do anything about column 4. People just don't think of it as amenable to medical intervention. The right way to think about the various types of way in which we can be sick is like this. All the columns are the same, the big black line is in a different place, that's the only difference. Really, the difference between column 3 and column 4 is purely semantic. The only difference is that the things in column 3 are the things that we have taken the trouble to give disease-like names to, and the things in column 4 we haven't. And we can say, well, okay, why would we do that? And you can kind of point to reasons, like, for example, maybe the things in column 3 are a little bit easier to delineate, to say whether you've got them or haven't got them. But that's a crappy reason for having any kind of distinction. Really, the essence of what I'm trying to get across here, and what I try to emphasize to everybody, is that this way of thinking about aging allows us to see 2 things. Number 1, it allows us to see that the attempt to treat these diseases in column 3 as diseases and try to eliminate them from the body is completely hopeless, because they're side effects of being alive in the first place. And number 2, that they should not be thought of as individual, separate phenomena. They should be considered as part and parcel of one phenomenon, as aspects of a single phenomenon, and therefore that the way we should be looking at doing anything about them is by a coherent, perhaps complicated, but nevertheless coherent, unified strategy. We can kind of talk about it like this. What I've said so far is basically described at the bottom here. Metabolism, in other words, the entire network of processes that keep us alive from one day to the next, causes damage to the body throughout life, even starting before we're born. And that damage is harmless for a long time, but then in late life, the damage translates into the various pathologies of old age. So, historically, this is what that popular view of the difference between aging and disease has resulted in. It's resulted in vast amounts of money being spent on an approach to treating those diseases of old age that cannot possibly work. Namely, diving in and trying to attack these symptoms as if they could be eliminated from the body like an infection. That's basically what geriatric medicine is. It's never going to work, because the damage that causes these pathologies is continuing to accumulate as an absolutely inevitable consequence of being alive, and therefore any geriatric medicine is bound to get progressively less effective as the patient gets older. It's worse than that, of course. There's also this minor problem, which is that quite a lot of things go wrong with us in old age. You're not supposed to be able to read this slide, don't worry. This is obviously a list of some of the things that people suffer late in life, and treating them one at a time is a bit of a losing battle. So, of course, I'm not by any means the first person to point this out. Probably for a century, some people have been noting that geriatric medicine was a bit of a fool's errand, and therefore that we ought to try to adopt a prevention is better than cure policy and dive in at an earlier point in the chain of events by essentially trying to clean up metabolism, trying to slow down the rate at which it creates damage in the first place. Of course, that would be great. It would postpone the age at which the damage reaches the abundance that causes pathology. Wonderful. A couple of problems there as well, though. First one is that the later you start a therapy of this type, the less effect it's going to have, because you've got all that damage that's happened before the therapies were initiated. And for those of us who happen to already be in middle age or older by the time the therapies are developed, there's not going to be very much benefit from these therapies. The other problem is perhaps even worse. It is that, just like pathology, metabolism is rather complicated. This, as I guess most of you know, is a simplified diagram of a small subset of what we know about how the body works. And it's rather hairy, really. It's completely insane to suppose that we could tweak this thing in such a way as to make it not do the thing we don't want it to do, namely creating damage, while at the same time somehow preserving all the things we do want it to do. It's just not going to happen. And of course, that's an understatement of the problem. The real problem is that this is a simplified diagram of a small subset of what we know about how the body works, which is completely dwarfed by all the astronomical amount that we don't know about how the body works, even ignoring all the stuff that we don't even know that we don't know. So, yeah, forget it, really. And that is a conclusion that was arrived at by pretty much everyone who studied the biology of aging through to the 70s and mid-80s or so. There was so much pessimism about the prospects of doing anything about the ill health of old age that it became distinctly unfashionable and frowned upon even to talk about intervention. Hardly anyone prominent in the field would do so. You would never ever mention actually doing anything about aging in a grant application, for example. Really, you know, gerontologists became like seismologists, people who study earthquakes. You know, they understand that the thing they study is bad for you, but they have no idea of how to do anything about them. So that was rather bad. And then, of course, everything changed when, in the late 80s and early 90s, a couple of labs found ways to extend the longevity of nematode worms, Caenorhabditis elegans, with simple genetic manipulations, just mutating single genes. And essentially what that achieved was a revival of this approach, of the approach of cleaning up metabolism. Because what it did was, it would slow down the rate at which damage would be accumulated. And we have to ask, well, okay, how come? What was wrong with what I just said about the complexity of metabolism? The answer is that it is possible for a simple intervention to have a very complicated but nevertheless coherent impact on a very complicated phenomenon, namely the accumulation of damage, if the body is already set up to actually have that response. In other words, if we have already evolved genetic machinery that is designed to respond to some environmental input and alter the way that metabolism works in a manner that slows down the accumulation of damage. And of course, we do have such a thing. We've known that for 50 years longer than we've had these genetic interventions. In the 1930s, it was first discovered that if you take a rat or a mouse and you give it less food than it would like, then it lives longer. And that's a really simple intervention. It's called famine, and it happens in the wild. So it's no surprise that that life has evolved ways to optimize metabolic priorities in response to the level of nutrient availability. And it doesn't take very much thinking about the pressures on evolution, the selective pressures involved, to see that it makes sense to alter those priorities so that you have your offspring at a stage in your life that is optimized so that they will have their own offspring and your genes will continue into subsequent generations. So this all made sense, and sure enough, the mutations that were discovered, and indeed the drugs that have since been discovered that actually have these effects, not only in worms, of course, but in pretty much every organism that's been looked at, are precisely the genes that are at the apex of this response to famine. They're genes that in one way or another detect the availability of nutrients and have downstream genetic and metabolic functions causing a whole wide variety of different changes that will slow down the accumulation of damage. So it shouldn't have been nearly so much of a paradigm shift as it actually was back in the early 90s. But anyway, this is what has dominated the field of the biology of aging ever since. And it's also given me a whole lot of trouble, because the dominance of this paradigm within modern biogerontology has led to a somewhat excessive over-optimism with regard to the impact that this could actually have. We see in these nematode worms, in which this phenomenon was first discovered, a very dramatic increase in longevity. One can multiply the longevity of these worms by a factor of 5 or 7 by this kind of method. And indeed, one can multiply the longevity of nematode worms by roughly the same amount by, sure enough, famine, by starving them at just the right point in their life cycle. Sure enough, if you do the same thing to mice or rats, genetic interventions of the same kind can again give about the same amount of life extension as you see for what you get with actual calorie restriction that has been tried since the 1930s. Unfortunately, the absolute amount that you get, the proportional increase in longevity that you get in mice and rats, is not a factor of 5. It's a factor of about 1.3, 1.4, something like that. And it turns out that if you look in longer-lived organisms, indeed this has now been done in monkeys, that the proportional increase is very much less still. So it's not looking all that good as a method for extending human lifespan. We'd be lucky, in my view, if we got even 2 or 3% increase in lifespan for humans from any kind of method like this, whether it be actual calorie restriction or a genetic or pharmacological intervention that emulates it. This is a rather disappointing conclusion, of course, and therefore it has been somewhat swept under the carpet by a large number of my colleagues, but that's life. However, it hasn't been swept under the carpet by me, and the result is that over the past 15 years, I and the people that I've been able to bring together under SENS Research Foundation have been pursuing a complementary approach, which is this. Rather than trying to slow down the creation of damage or to combat the consequences of damage, instead, we propose to go in and periodically repair the damage itself, to go in and separate, essentially uncouple this process from this process by allowing the damage to be created, but then removing it, not necessarily completely, but removing lots of it, so that it does not reach the threshold level that causes pathologies to emerge. And of course, this is what we already do with inanimate objects, coming back to my point that the human body is merely a machine. Here is a car that's about 100 years old, and the critical thing is it was not designed to last 100 years. It was probably designed to last 10 or 15 years. Why has it lasted so long? Maintenance, preventative maintenance, periodic removal of initially harmless damage, like rust, before that damage reaches a level that makes the doors fall off. That's what we do successfully to extend the fully functioning, healthy longevity of a simple man-made machine far beyond what it was designed to have, and indeed essentially indefinitely. And there's no reason in the world why we shouldn't be able to do the same thing, in principle, with a much more complicated machine such as the human body. So what does this actually amount to in practice? What does it mean when we come down to the specifics of the biology of damage and its repair? What it means is that we have to actually, first of all, characterize what the damage is. We have to look at, let's go back again to my 25-year-old person and my 45-year-old person. We have to look at what's different in the bodies of those two people that could account for the fact that the 45-year-old person has so much less time to live. And that's where the whole paradigm, the whole research agenda of SENS Research Foundation originally came from, because I realized sometime back that all of the various types of damage that we know about that seem to be contributing to the ill health of old age can be classified into this very manageable number of categories, just 7 categories. I won't go through all of these now, not least because most of you have seen this slide before or something like it. But just for illustration, the top one here, cell loss. What's that? It's simply cells dying and not being automatically replaced by the division of other cells. It's a perfectly straightforward phenomenon. And of course, we know what the fix is. We know what the preventative maintenance approach is to this, which is stem cell therapy. Putting our own cells into the body, putting new cells in that will divide and differentiate to replace the cells that the body is not replacing on its own. And the same kind of very straightforward, down-to-earth, concrete approach can be described for all of these other things. The reason why SENS Research Foundation exists is that the research that's being done by other people around the world to develop these other preventative maintenance methods against these other types of damage is at a very much less advanced stage than we see with stem cell research. So, very few people really understand the importance of these types of research even. I want to spend a couple of minutes just emphasizing the relationship between damage and pathology, because of course that's really where the rubber hits the road. That's what it's all about. I've told you in the abstract that damage accumulates and that eventually causes pathology, but really it's much easier to get your head around that concept if you can actually see what the relationship is in specific examples. Sometimes the relationship is very straightforward. So cancer is probably the simplest one of all, because one of our categories is division-obsessed cells. In other words, cells that are in a state where they divide when they're not supposed to, and therefore you get too many of them. That's essentially the definition of cancer. But in most cases, the situation is more complicated. So there's a lot of things that can go wrong with the heart as you get older, and they turn out to be caused by different types of damage. So atherosclerosis, the #1 killer in the Western world, which causes heart attacks and strokes, that is a process of accumulation of waste products inside the cell. That's what intracellular junk is. There's a particular type of waste product, a type of oxidized cholesterol, that is the main problem. Arteriosclerosis, the hardening of the arteries, which leads to high blood pressure in the elderly, is caused by this bottom one down here. Stiffening, loss of elasticity of this structure called the extracellular matrix, this lattice of proteins that holds our tissues together and gives them their physical properties. Then there's amyloidosis, which has, over the past few years, been appreciated as the #1 killer of really, really old people, people over the age of 105 or so. That's something that happens in the heart. It's rather like the amyloid that we see in the brain in Alzheimer's disease, though it's made of a different material, and it's a type of waste product that accumulates outside the cell, in the spaces between cells. Then finally, there is actually cell loss in the heart as well. There's a particular population of cells in the heart called pacemaker cells, which do not maintain their number during life, and those cells are rather important because they are the cells that mediate the communication between the heart and the brain, and that communication is what actually makes the heart beat. The heart only beats when the brain tells it to, and these cells, if they are too few in number, simply do not transmit that signal properly, so the heart can simply stop as a result of lack of pacemaker cells. If we look at Alzheimer's disease, going to the brain now, 100 years ago, when Alzheimer's disease was defined, the definition was the combination of these two things down here, a type of garbage inside the cell called tangles and another type outside called plaques. And now, of course, we also know that there's a lot of cell death in the brain in Alzheimer's disease, and those dead neurons are not replaced. So again, it's a complex phenomenon, but the key thing here is I'm not making any of this up. None of this is speculative. All of this is really well-established, uncontroversial facts in terms of the relationship between damage and pathology. There's plenty of stuff not known about the details of how the damage translates into the pathology, but that's not what this is all about. What this is about is simply what damage, what the types of damage are that cause the pathology. And the key thing I want to emphasize finally in this section is that the same is true for those other aspects of aging that are in column 4 of my original table, the ones that people don't think of as diseases. It's the same deal. If you look at any of these things that go wrong, like decline of the immune system, for example, you can trace them back to the same bunch of types of damage. So that's why this kind of classification seems to be really rather promising. And I've been challenging people, indeed, to challenge it, to come up with reasons why it's not exhaustive, why it's not thorough, why there should be a category number 8 or number 9. And I seem to be getting away with it. I've been talking about this kind of thing for more than a decade now, and people have had a go and they've failed. So that's nice. So I'm going to spend the rest of my time, because I know a lot of you are quite familiar with everything I've said so far, I'm going to spend the rest of my time talking about some recent work of SENS Research Foundation itself, and some actual data, some updates on our project. As actually, these slides have been made in order to give a comprehensive overview, which I don't have time to give, so I'll skip through some of this rather quickly, but I'll highlight some of the most interesting data that we've been able to generate lately. So we don't do much in the top category, cell loss, simply because so many other people are doing stem cell work. But we do have one project at this very prestigious institute in North Carolina, one of the best-known regenerative medicine outfits in the world, actually. And we're looking at creating a new thymus. The thymus is an important part of the immune system. It shrinks and goes away during aging, and that eventually results in our not having as good an ability to respond to new infections as we used to have. We want to make a new one, and we're using a method that's also being used called decellularization, that's also being used across the world for various other organs, but nobody seems to be trying it for the thymus, so we are. For cancer, we are focused on the telomere. Many people like to say these days that cancer isn't really one disease, it's a whole collection of different diseases, and of course that's true and false. In other words, yes, different types of cancer have different characteristics, but they also have common characteristics. And the single characteristic that all cancers most reliably have is that they all have, in human beings, some way to extend the telomere, the end of the chromosome, to compensate for the telomere shortening that happens during cell division. Cells that get into a cancerous state but they don't turn that on will not kill you, because the number of cell divisions that those cancers will be able to undertake before they fade away on account of not being able to extend their telomeres is insufficient for the cancer to grow enough and to metastasize enough to kill you. So we're working on that, and one thing we're doing is trying to figure out the genetic basis of the second way in which cancers turn on telomere elongation. Everyone understands the first one, this enzyme telomerase, and we want to understand the other one, which hardly anyone is working on. It's called ALT. We're doing that at our research center in California. Then we're also looking at ways to alleviate the side effects that would occur if we were to suppress the elongation of telomeres indiscriminately across the whole body, and to do it really, really powerfully. The side effects that would occur would be a failure of an eventual failure of cell division in the stem cell compartments of rapidly renewing tissues, such as the blood and the gut lining and the epidermis. And we have a project at this same institute, Wake Forest Institute, looking at the gut, looking at a very remarkable phenomenon whereby the gut lining is replenished from mesenchymal stem cells that are circulating in the bloodstream, and trying to improve the efficiency of that system. And then we had a project that just very recently completed in China, looking at the blood and determining whether we could rescue the stem cell function of blood in mice whose stem cells in their blood had become compromised as a result of telomere shortening. And the long and short of it is it works perfectly. Essentially, without going into too many details, what we have here in the red line are mice that are living less long than normal because their telomeres are too short and they don't have any gene for telomerase, so they end up getting anemia. And these mice were rather difficult to create because we had to make sure that they only got anemia, that they didn't have problems with their digestion as a result of having also critically short telomeres in their gut. But it turned out that we worked with a group originally from Germany that were doing exactly that, that could do exactly that, using a combination of two mutations. Turns out that if you take these mice and you give them normal hematopoietic stem cells, then sure enough they live longer. It's a relatively modest increase in longevity because eventually these mice do get problems with their digestion as well, which of course are not rescued by this hematopoietic stem cell bone marrow transplant. But it's enough to be statistically significant, so that's fine. And that's the green line. And the important one, of course, is the black line, which is when we give a bone marrow transplant using stem cells that do not have the capacity to increase their telomere length, but they do start out with nice long telomeres, so that they can divide a nice number of times anyway. And as you can see, the rescue is every bit as good. The black and the green line are on top of each other. Which shows that, for this purpose anyway, there is no need to worry about possible other roles for telomerase in addition to increasing telomere length, for example. So we're very happy about that. It's not been published yet, of course, but we only just recently concluded the study, but it's definitely a success. We're worried about cells that won't apoptose as well. People don't often think about this. They think, well, okay, why would you ever want cells to die? But it turns out that there are areas in the body where that's very important. Whoa, it's already 5 to 12. Okay, I lied when I said I'd be finished by 5. The immune system is the most important one, probably. The immune system, it's absolutely vital that cells should die when an infection has been eliminated so as to make room for the proliferation of other cells in response to the next infection. And we're working on a system of a kind of advanced version of dialysis at Berkeley in order to be able to remove such cells from the body when they have got into a state where they're not dying spontaneously. We're also working on drugs and indeed gene therapy methods to get rid of the cells that accumulate elsewhere in the body as a result of various damage to their DNA, which again get into a state which is what we call death resistant. Now, with regard to what happened there? Okay. With regard to molecular garbage inside the cell, we have a couple of projects that we feel are very important. One of them is with regard to macular degeneration, the #1 cause of blindness in the elderly, about which you heard yesterday, of course. And the other one is with regard to atherosclerosis, the #1 killer in the Western world. We're working on both of them, and our method is to identify enzymes in other species, typically in bacteria, that are able to break down the substances which cause these diseases. In respect of macular degeneration, we are making progress, but we haven't really got anything exciting to report yet. But with respect to atherosclerosis, we're very happy with the situation. So this is the problem we're trying to address. This is the early stage of atherosclerosis, where a perfectly healthy white blood cell goes into the artery wall in order to clean stuff up, and it gets poisoned by oxidized cholesterol. The part of the cell that gets poisoned is this thing, the lysosome, which eventually fills up with fatty stuff, and then the whole cell starts to fill up with fatty stuff, and the white blood cell turns into this kind of undead thing, which people call a foam cell. And that's the first stage of atherosclerosis. So we've been able to do this. We've been able to find bacteria that can break down the #1 target here, something called 7-ketocholesterol, and we've been able to find the gene and then the enzymes that they use to break it down. And then, the hard part, we've been able to modify those enzymes so that they work in human cells. Specifically, this enzyme we're using, which is a cholesterol oxidase. So what this graph is showing is simply that if you have a completely astonishing amount of this toxic molecule, then all the cells die anyway. But if you have more modest concentrations, then the right-hand bar in each of these groups, as you can see, is higher than the others. That simply shows that the cells are happier, they can survive better in the presence of this toxin than any of these negative controls, such as a cell that doesn't have the enzyme, or it has the wrong enzyme, or the enzyme is not targeted to the right part of the cell. So this was published 2 years ago now, and in that time we've been working to try to transfer this technology to the right type of cell. This was just done in fibroblasts, and we need to have it working in macrophages. And then, of course, we're moving on to mouse models. So that's making good progress. I'm going to just whip through this and just talk about the last one, because this is the one that I've got data for. The right one? No, the wrong one. So I'm actually going to talk about the middle one, extracellular waste products. So I mentioned that amyloidosis is the #1 killer of people who are extremely old, supercentenarians. And we want to use the immune system to get rid of the material in question. This has already been done very successfully in the case of Alzheimer's disease. People have found that you can vaccinate against the amyloid that accumulates in the brain, and the macrophages of the brain, microglia, will engulf this stuff and get rid of it, and then destroy it within the lysosome. So that's very helpful. We've actually been exploring a slight variation on that theme in respect of the amyloid in the heart. We've been looking for antibodies that, instead of causing phagocytosis of the material, they protect the cells from the material by being neutralizing. Essentially, they mean that the material doesn't poison the cells anymore. And we've been also looking at a very astonishing technology that was developed by a group in Houston, which involves antibodies that don't just bind to their antigen, they actually chop it up. They're proteolytic. And we've been able to get very promising data in both cases. Here's what we've got with regard to neutralizing antibodies. This is just in culture. We've got cells that are perfectly happy normally, and their viability is halved by the presence of this amyloid. And when we apply the lead antibody so far, that viability is completely restored to the original levels. So that's extremely good news. With regard to the proteolytic antibodies, the difficulty here is that we want to be selective for the type of protein that is aggregating into this amyloid form. The protein in question, like all proteins, is made for a purpose. It has an actual physiological function in the body. It's called transthyretin. It shunts a particular hormone around the body, and it does so in a form that's a tetramer. 4 of the molecules bound together in a certain conformation. That tetrameric form does not aggregate into amyloid. But when it misfolds in an interesting way and doesn't form a tetramer, and you just have a monomer, then this is the dangerous form. And we've got an antibody that can completely destroy, in no time at all, the monomer and doesn't do anything to the tetramer. This was published in the Journal of Biological Chemistry a few months ago, and it's probably our highest profile published result so far. I'm not going to have time to go through any of that. I will stop and let you ask questions, or alternatively go and check out. Thank you.
Thank you very much. Any questions? Let's start right here.
So let's assume that one of your projects, for example, an enzyme for breaking up intracellular junk is almost there. So it's on the threshold of being turned into a drug. So how would you proceed? Doesn't it require huge amounts of money nowadays to turn something into a drug?
So, yeah, I mean, our goal, as we often describe it, our mission as a foundation, is to create a rejuvenation biotechnology industry. So we definitely seek to take these things forward into the private sector and to take them and to get the investment that would be required to go through the standard clinical trial process. Yes, it's a huge amount of money. I agree. I guess that the main reason we're not too worried about that is because huge amounts of money are already spent in the development and eventual delivery of drugs that work far less well than the drugs that we are producing. So if we take the case of the atherosclerosis example here, we've got statins now. Statins are one of the most successful drug families in the world, and they really don't work very well because they attack the problem at the wrong point. They work by diminishing our ability to synthesize actual normal cholesterol, which is a vital molecule, so there's only so much you can take.
Thanks. I have a comment, actually criticism, and a question. So the comment is you said that aging of human beings is like aging of inanimate objects, which is one of those things that makes me cringe, and I have to say I disagree with, for 2 reasons. First of all is that inanimate objects do not have the ability to self-repair. As you know, our cells can repair DNA, proteins, etc., which inanimate objects cannot. And the second reason is that inanimate objects, with time, do not improve in function or complexity, as it happens in humans during development, which goes from a tiny cell to a very complex. So there is a comment which...
Okay, well, let me address that first before your question. I won't forget that you have a question.
Okay.
So, of course, both of the things you've just said are true. But I don't think that either of them affects the validity of the analogy for these reasons. Let me deal with the second one first. So yes, absolutely, the body creates itself from a single cell all the way up to an adult. And that's a remarkable phenomenon, but it's a phenomenon that's completely separate from the accumulation of damage, which also happens starting from the single cell, even before we're born, but which happens as an unprogrammed side effect of what the body is programmed to do. So I don't believe that the way in which the body is built, the fact that it builds itself rather than having to be built by design, by other machines, is a relevant difference between living and inanimate objects. With regard to the other one you mentioned, namely the fact that we have all this built-in self-repair, yes, that's a big difference. But it's a difference that makes my point stronger, because it means that we have less to do to augment the existing programmed repair that we already have. If we didn't have that repair, if we were more like inanimate objects, then the problem of actually developing that medical repair would be much harder than it actually is.
Right, so I think there's a difference in then the way how you're going to intervene, or you're going to repair it, what is an inanimate object.
No, I don't think so. The repair is the same. It's just that we have to do less of it because so much of it is happening without our efforts.
Okay, we can discuss in more detail. My question, which I think would be of general interest to people in working cryonics, is very simple. When do you think we're going to end aging? Do you think it's still going to happen in our lifetime, or in your lifetime? And if you do think it's going to happen in your lifetime, what do you expect to see, say, 5 or 10 years from now, to occur to know we are on track to that?
Yeah, I am still very optimistic. So I started talking about timeframes for the development of all of this about 10 years ago. And what I said back then was that I thought it would be about 25 years before we got all of this working, but with 2 caveats. First caveat was, I said, I think we've got a 50-50 chance of getting there within 25 years, but for any technology that's that far away, the speculation index is extremely high. It's definitely at least a 10% chance that we won't get there for 100 years. But that doesn't matter, because a 50% chance is quite enough to be worth aiming for. The second thing I always used to mention, and always still do, is that it's all subject to funding, and that we're not going to get there without actually trying. So I suggested that with appropriate funding, it would probably only take 10 years before we got to an equivalent kind of point in the case of mice, that we could demonstrate, like taking middle-aged mice and perhaps doubling or trebling their remaining healthy lifespan by a series of interventions of this nature. And of course, it's 10 years on now and we haven't got there, but we haven't had the money. And if I look back and I ask, well, how much progress do I think we would have made with the money that has actually been forthcoming, I think I got it pretty much spot on. I think we've got about 1/3 as fast as we could have gone, and that's about what I would have expected. So I would not say that I was either over-optimistic or over-pessimistic 10 years ago. What that means for the future is we've still got a job to do to bring more money in, but of course we all know that that's accelerating really fast with the entry into the field of entities like Calico, for example. So I feel very much more optimistic about the funding situation and therefore about the timeframes than I would have had any right to do 10 years ago.
I'm no molecular biologist. I'm only anatomist, but I can give you an example of the complexity of ending aging, namely the kidney. You have a loss of glomeruli, and there is, during embryonic development, a few days where 3 tissues come together, and only at this period a glomerulum can be formed. If you have an infection by streptococci, you can lose it, and it's never replaced, only by a scar. Thus, you would to have engineer all these tissues and bring them together, and you must degrade this scar. And I think your lifetime will not allow to end aging if you go from one organ to the other and find different problems in each of the organs.
Yeah, so I think that's actually a great example to bring us down to earth about the complexity of all this. If I thought that this was simple, I wouldn't be talking about a 50% chance of getting there in 25 years. I would be talking about a 50% chance of getting there in 5 years. So I completely appreciate and agree with what you say. These are very daunting problems. However, if we look at every single step of what you say about the kidney or about what you could say about any other organ, we can see specific and detailed routes towards making this happen. We know of many cases now where scar tissue can be eliminated by the action of, especially of stem cells, in the nervous system is the place where this has been studied the best, and this is certainly a major area of research at this point. So I don't believe it's a problem on the scale of we just have no idea what direction even to try. And similarly, with regard to the complexity of the glomerulus and these three types of cell that you mentioned, yeah, they do have a common precursor cell, of course, progenitor cell. And the understanding that we have of the lineage of the progenitor and stem cells, starting from the zygote and the totipotent stem cell all the way through to various differentiated cells, that knowledge is burgeoning and increasing all the time. Of course, not only the knowledge of what happens, but also of how to stimulate it to happen so that the right stem cell can be introduced into the right place in the body. A great example of this is Parkinson's disease. I'll just mention this story for a moment because it's quite salutary. About 20 years ago, in Sweden, people said, well, okay, Parkinson's disease, it's the classical aspect of aging that can most easily be addressed using stem cells, because basically it's driven entirely by the loss of one particular type of cell in one particular small part of the brain. It sounds like a really easy thing to do. You just got to put in the right kind of stem cell. And they had a go. They took some fetal stem cells from the right part of the brain, and they injected them. And occasionally it worked, but most of the time it didn't work. And the same idea was tried in America a couple of times, and it didn't really work there either. So most people kind of gave up on the whole thing. They said, oh, it's not going to work, it's not going to work. But some people paid attention to the people who did actually respond in the initial trial. And they're still paying attention to those people. There are a few people walking around today who had one stem cell treatment in nearly 20 years ago, and they've had no L-DOPA or any other kind of treatment for their Parkinson's ever since, and they're still completely free of symptoms. So now people are taking on board the fact that this can work, and that now that and that the reason it usually didn't work was that the wrong type of stem cell was being transplanted. Cells that simply did not actually differentiate into dopaminergic neurons, the right type of neuron. So now, of course, we know far more about how to determine whether a stem cell is the right cell and how to manipulate them and so on. And sure enough, new clinical trials are starting up, and people are very optimistic. And I would say at this point, actually, I would say there's a 50% chance that within as little as 10 years, we will be able to say that Parkinson's disease is cured. So this is just an example of how much things can change as a result of knowledge that is initially rather, esoteric, and we don't really see how relevant it's going to be, but things can move awfully fast after that knowledge is applied.
Well, thank you very, very much.